Cancer cells possess a remarkable and dangerous ability. They can escape one of the most important limits built into normal human cells, the limit on how many times a cell can divide. Normal cells are not designed to reproduce forever. Yet during cancer development, some cells bypass the biological barriers that normally stop division. They acquire what scientists call replicative immortality, the first hallmark of cancer. Let us begin with the first image. Every organ in the body has a defined size, structure, and function. This organization is maintained because cell division is tightly controlled. Cells divide when new cells are needed for growth, tissue renewal, or repair, and they stop when enough cells have been produced. This regulation occurs through the cell cycle. During G1, the cell grows. During the S phase, it copies its DNA. During G2, it prepares for division, and during mitosis, or the M phase, it separates into two daughter cells. But the cell cycle is not an automatic wheel that turns without supervision. At several checkpoints, molecular systems inspect whether the cell is ready to continue. Is the DNA intact? Was it copied correctly? Is the cell receiving the appropriate growth signals? If serious damage is detected, division can be delayed or stopped. This control preserves tissue balance. Without it, cells could continue multiplying until they formed abnormal masses, disrupted tissue architecture, compressed surrounding structures, and interfered with organ function. Cancer develops when these regulatory systems begin to fail. Mutations may activate growth-promoting genes or disable the genes that normally restrain division. The result is uncontrolled proliferation and the gradual formation of a tumor. However, uncontrolled growth alone is not enough to sustain a cancer indefinitely. A cancer cell must also overcome another powerful defense, the natural limit on the number of divisions available to most normal cells. Now let us move to the second image. One of the most important discoveries in cell biology came from Leonard Hayflick and Paul Moorhead. Their experiments showed that normal human cells grown in culture do not divide forever. After a finite number of population doublings, many cells permanently stop proliferating. This phenomenon is commonly associated with the Hayflick limit. The precise number varies with cell type, donor, age, and laboratory conditions, but classic human fibroblasts often divide approximately 40 to 60 times before reaching this limit. When cells stop dividing in this stable manner, they enter a state called cellular senescence. A senescent cell is not necessarily dead. It remains alive and metabolically active, but it no longer progresses through the normal cell cycle. In laboratory cultures, these cells often become enlarged and flattened, producing the characteristic appearance sometimes described as resembling a fried egg. Senescence acts as a natural barrier against cancer. A cell that has divided repeatedly, accumulated stress, or developed potentially dangerous damage is prevented from producing additional descendants. One of the major signals that drives replicative senescence comes from telomeres, the protective structures at the ends of chromosomes. With repeated cell division, telomeres progressively shorten. When they become critically short or dysfunctional, the cell recognizes its chromosome ends as damaged DNA. This activates protective pathways involving proteins such as p53 and Rb. The cell cycle is stopped, and the cell may enter senescence rather than continue dividing with unstable chromosomes. Normal dividing cells, therefore, have a limited reproductive lifespan. They grow, divide a finite number of times, and eventually stop. This protects tissues from endless cellular expansion. Cancer cells, however, must somehow break through this barrier. Let us move to the third image, where one of the most famous examples in biomedical research shows what happens when cells overcome this limit. In 1951, cells were collected from the cervical cancer of a woman named Henrietta Lacks. These cells produce the first widely used immortal human cell line, known as HeLa. Most normal cells eventually stop dividing in culture. HeLa cells did not. Descendants of those original cancer cells have continued proliferating for more than 7 decades and are still used in laboratories throughout the world. Calling HeLa cells immortal does not mean that an individual cell cannot die. It means that the population can continue reproducing under suitable conditions without reaching the usual replicative limit. The contrast with normal cells is striking. Normal cultured cells initially grow and divide, but their telomeres gradually shorten, and many eventually enter senescence. HeLa cells maintain their telomeres and bypass important senescence pathways, allowing continuous proliferation. Several molecular changes contribute to this ability. HeLa cells express the enzyme telomerase, which helps preserve chromosome ends. They also originated from a cervical cancer driven by high-risk human papillomavirus. Viral proteins disrupted the p53 and Rb tumor suppressor pathways, weakening two important barriers against uncontrolled division. HeLa cells have contributed to major discoveries in cancer research, genetics, virology, drug testing, vaccine development, and space biology. However, their history also raises an essential ethical issue. Henrietta Lacks' cells were collected and propagated without the informed consent expected today. Her story has therefore become central to discussions about patient rights, research ethics, and the use of human biological samples. To understand why HeLa cells and many other cancer cells can continue dividing, we must now examine the chromosome ends more closely. Let us continue with the fourth image and focus on telomeres. Telomeres are repetitive DNA sequences, together with protective proteins, located at the ends of chromosomes. In humans, the repeated sequence is TTAGGG. A useful analogy is the plastic tip at the end of a shoelace. Just as that tip prevents the lace from fraying, telomeres prevent chromosome ends from being mistaken for broken DNA. They also help protect chromosomes from degradation and inappropriate fusion with one another. However, telomeres short whenever most normal cells divide. This occurs because DNA polymerase cannot completely copy the very end of a linear chromosome, particularly after the final RNA primer on the lagging strand is removed. This limitation is known as the end-replication problem. A small amount of telomeric DNA is therefore lost during each division. At first, the shortening causes no immediate harm because telomeres act as protective buffers, but after many rounds of division, they may become critically short. The chromosome ends then trigger a DNA damage response. And if normal protective pathways are functioning, the cell stops dividing and enters senescence. Telomeres can therefore be viewed as part of a molecular clock that limits the proliferative lifespan of many normal cells. This creates a major obstacle for cancer. A tumor may need to produce billions of cells. If its telomeres continued shortening with every division, the cancer population would eventually reach a crisis in which chromosome ends became unstable and continued growth could no longer be sustained. To become immortal, cancer cells must maintain their telomeres. The most common solution is telomerase. Telomerase is a specialized enzyme containing both a protein component and an RNA template. It uses this template to add new TTAGGG repeats to the ends of chromosomes, replacing some of the telomeric DNA lost during replication. In effect, telomerase helps maintain or reset the cell's molecular clock. Most normal adult somatic cells have little or no detectable telomerase activity, although important exceptions include germ cells and some stem or progenitor cells. By contrast, telomerase is active in approximately 85 to 90% of human cancers. A smaller group of tumors maintains telomeres through another pathway called alternative lengthening of telomeres, or ALT. Now let us move to the fifth and final image, where the differences between normal and cancer cells become clear. In a typical normal somatic cell, low telomerase activity means that telomeres gradually shorten with repeated divisions. Eventually, critically short telomeres activate senescence and proliferation stops. In many cancer cells, telomerase is reactivated. The enzyme extends chromosome ends and keeps telomeres above the critical threshold. This allows the cancer lineage to continue dividing far beyond the normal limit. Importantly, telomerase alone does not usually transform a healthy cell into cancer. Cancer requires additional genetic and epigenetic alterations affecting growth control, cell death, DNA repair, immune interactions, and other biological systems. Telomerase is better understood as an enabling mechanism. Once a cell has acquired cancer-driving changes, telomere maintenance allows that abnormal clone to keep expanding without exhausting its replicative capacity. This mechanism may also support cancer stem cells or other tumor-propagating populations. These cells can sustain tumor growth over long periods and may arise from transformed stem cells, progenitor cells, or cells that acquire stem-like properties during cancer evolution. Advanced and metastatic tumors also commonly show telomerase activity. This does not mean that telomerase directly causes metastasis. Instead, it gives cancer cells the long-term proliferative capacity required to expand at the original site and establish new populations in distant organs. Senescence itself also has a complex role in cancer biology. At the cellular level, it is an important tumor-suppressive mechanism because it stops damaged cells from dividing. But senescent cells can release inflammatory molecules, growth factors, and enzymes into their surroundings. This collection of signals is called the Senescence-Associated Secretory phenotype, or SASP. Under certain conditions, these signals may alter nearby tissues and contribute to aging or tumor progression. Because telomerase is active in most cancers but low in many normal tissues, it has attracted attention as a possible therapeutic target. In principle, blocking telomerase could allow cancer cell telomeres to shorten until continued division becomes impossible. However, this approach is challenging. Telomere shortening may require many cell divisions before producing an effect. Some normal stem cells also use telomerase, and certain tumors may switch to alternative telomere maintenance mechanisms. Nevertheless, telomerase and telomere biology remain important areas of cancer research. The central lesson is that normal cells live under several layers of protection. Their cell cycles are controlled, their DNA is inspected, their telomeres shorten, and their capacity to divide is limited. When damage or excessive replication is detected, they can stop through senescence. Cancer cells progressively dismantle these barriers. They disrupt cell cycle checkpoints, bypass senescence, and activate telomere maintenance mechanisms, most commonly telomerase. Together, these changes give cancer cells the potential for sustained, long-term proliferation. This is the true meaning of replicative immortality. It does not simply mean that cancer cells divide rapidly. A rapidly dividing cell may still have a limited lifespan. Immortality means that the cancer lineage can continue reproducing without reaching the natural division limit imposed on most normal cells. From the controlled cell cycle, to Hayflick's discovery of senescence, to the extraordinary history of HeLa cells, and finally to the molecular biology of telomeres and telomerase, one principle becomes clear. Cancer is not simply excessive growth. It is the progressive escape from the biological systems designed to place limits on growth. And by understanding how cancer cells achieve immortality, researchers may uncover new ways to restore those limits and stop tumors from growing.